All right, we're going to go ahead and get started. Good morning, everyone, and welcome. My name is Brianna Widener, and I'm the Outreach and Education Specialist for the Washington Invasive Species Council. And we are an interagency organization tasked with managing invasive species priorities around the state. We bring together different agencies to try to make our efforts more effective by working together. So today, we have one of our partner agencies here. Joining us is Rachel Baumberger. Rachel is a plant diagnostician at the Washington State University Plant Pest Diagnostic Clinic, and today she's going to be talking about tree threats to the Pacific Northwest, including the biology, identification, and lookalike issues. She's also going to cover some detection methods and a response for some of these tree troubles. So at this point, I'm going to pass the mic over to Rachel for the Tree Threats, Xylella fastidiosa, and Basics of Borer Damage webinar. Hope you enjoy it.
All right, thank you so much, Brianna. So my name is Rachel Baumberger. I work at Washington State University on the Pullman campus with the Department of Plant Pathology, and I run the Plant Pest Diagnostic Clinic. So today I'm going to be talking about tree threats. So if you're on this webinar, there's a good chance you really like plants. And trees are extremely important to both us and our environment. We've seen studies that show that it helps actually people's physical well-beings as well as mental well-beings, but unfortunately they are at risk. There are many things that can cause a tree to struggle. They can be human related. I myself cause a lot of what I like to call Rachel blight on trees, and I'm sure many of us have come across trees with 8-foot canopies and 4-foot concrete planters. I know I have, at least. We can also have other environmental issues or non-living abiotic things such as drought or nutrient stress, or sometimes it can be damaged from living organisms such as a disease or a pest. Today I'm going to be talking about a particular disease, and then I'll finish up by talking about borers as a group of organisms that can cause damage to trees. All right, now with that, let's move into our first topic, bacterial leaf scorch caused by Xylella fastidiosa. Now, full disclosure, I am a plant pathologist, so I study plant diseases, so I might get really excited talking about certain aspects of the disease, so please forgive me for that. As the name suggests, this is a bacterial disease, but this pathogen has caused different diseases important to the U.S., including things like Pierce's disease on grape, citrus variegated chlorosis on citrus, and it has been the cause of olive quick death in the EU. And it's a problem on many different species of plants. So it is quite an interesting pathogen that we've been having to deal with here. So brief history on Xylella. So it was actually first found in California vineyards in the 1890s. So, over a century ago, we found Pierce's disease. It was actually found by one of the first formally trained plant pathologists, Newton B. Pierce. He was the state plant pathologist for California. So he found this disease in 1892, and what was happening is they were seeing destruction of over 14,000 hectares of grapes planted in the area. And it's caused by the Xylella fastidiosa subspecies fastidiosa. We're going to talk more about this subspecies or strain caveat of this disease. And what he noted is that there was a pattern in the field that was more typical of a virus rather than a bacterial infection. Often with bacteria, we see a lot of really wet lesions, pretty extensive rotting, and often can smell quite bad due to the pathogen. It's also important to note that around this time in Georgia, peach growers were starting to see stunted trees that they call pony trees, kind of like a pony keg as a reference, that were producing fruit that was undersized and unmarketable. They actually called this phony peach disease. It was only until later that we found out that these two diseases were actually caused by this same pathogen. But more on this in a moment. It's definitely an interesting organism because, again, we couldn't figure out exactly what was going on with this disease because the pattern in the field looked very typical to virus, and it wasn't until about the 70s that we determined that it was truly a bacteria. And it wasn't until the 80s that we were able to conclusively prove that it was this bacteria causing the damage. So a bit about Xylella fastidiosa. So Xylella fastidiosa is actually the only species in this genus. And if you speak plant biology, Xylella really kind of translates to xylem. The xylem is actually the part of the plant that moves the water transport. So a very critical part for the plant. This bacterial pathogen only exists in these xylem sieves or water conducting tissues. So that's where we derive the name from. Fastidiosa comes from fastidious. So this is a really picky eater. It only likes to be in certain areas of the plant, and we don't find it outside of the area. It's actually the only bacteria that we currently know of right now that likes to exist in the xylem. Its picky eaterness, if you will, is actually what caused it to be misdiagnosed and undiagnosed for so long, because most of the time in a laboratory setting, what we do is we actually culture particularly fungal and bacterial pathogens out on artificial food. We call it media. And so we take pieces of the tissue and hopefully try and coax the pathogen out into this artificial growth medium. That way we can identify it. So without us being able to get this bacteria to show up in a culture plate, that kept kind of suggesting that it was virus, so that it was fitting virus in the field, fitting virus in the lab. So it took us quite a while to actually find a particular type of false media or false food that allowed the pathogen to grow. So it does not always reproduce in its host. So this bacteria does not always start reproducing and copying itself or dividing. And this means that we don't end up clogging the xylem vessels. Hopefully y'all can see my cursor here, but here we have the xylem vessels, and you can see the bacteria kind of clogging up the different areas. So if the bacteria is not dividing and not replicating, we don't get a clog of the xylem vessel. So we don't have the resulting symptoms or the leaf scorch that is quite typical for this disease. So that means we can have asymptomatic plants. And I think many of us here can guess what the issue of asymptomatic plants will end up being. Now, there are 6 different strains of this particular pathogen. This is a really complicating factor because we have strains that are likely endemic, especially to the Americas, both North, Central, and South America, but we also have strains that are not, that they haven't been to, they can be introduced into new areas. They can have new combinations of hosts that were used to one particular strain, but not another. And this is where we can get into problems, especially invasiveness issues with this particular disease. So this can definitely be quite the complicating factor. And then next, this is a vector transmitted disease. So very similar to viruses or spiroplasmas or another type of bacteria called a phytoplasma, we rely on the vector to move this pathogen around. Oh, I want to show one more thing. Again, I told you I was going to be overly excited about this particular pathogen. So this is what one of the bacterial cells looks like for Xylella. Now, it doesn't have flagella, which are these kind of whip-like structures that move the pathogen around. Instead, it has these little kind of ridge-like structures, and these ridge-like structures actually allow the bacteria to adhere to the xylem vessels and kind of stick in there really well. And that's what can make them very problematic, in addition to the fact that they're clogging up the xylem tissue, is that they're really good at staying put in that tissue. For those of you who are particularly curious or like bacteria, this is a gram-negative and does require oxygen to live bacteria. Okay, now on to the virus vectoring. So this bacteria exists in the xylem, so we have to have a xylem-feeding insect to transmit it. Now, unfortunately or fortunately, these do exist. Particularly, we have the sharpshooters in the family Cicadelidae. Again, they have piercing, sucking mouth parts. So again, very, very typical for pathogen vectoring organisms, because they're able to stick their little stylet into the affected tissue, feed on it, and then move to an uninfected plant, thereby inoculating it or introducing that disease to that area. If there's a low level of bacteria, sometimes we see low levels of transmission. So these are sharpshooters and leafhoppers. They are the main vectors in regards to moving the pathogen in the Americas. Now in the EU, what we've been finding is spittlebugs. So this organism right here that oddly enough I find quite cute in a way. If you've ever walked around and seen those kind of foam showing up on your plants unexpectedly, that is a spittlebug that is deep inside there. What we're looking at here is a little nymph. So these belong to the family Serpidae. Now, an important thing about these vectors is they're actually present across the globe. So this can be quite problematic when it comes to the spread of the disease, because if we can introduce an infected plant into a new area, there's likely going to be potential vectors because both the sharpshooter and spittlebug family are present across the globe. So again, this is a concerning issue when we talk about the spread of this particular disease. Now, this organism right here, glassy wing sharpshooter, is actually an invasive bug itself. It was accidentally introduced into California in the 1990s and started spreading Pierce's disease further than it had originally been transmitted by this blue-green sharpshooter. The blue-green sharpshooter tended to be in riparian areas, but when the glassy wing sharpshooter moved in, all of a sudden this organism can start moving away from the riparian areas. So more plants were starting to become infected due to this organism. So not only can we have this pathogen and its various strains be problematic and invasive, we can have the vectors be invasive as well. So we're going to move on to the symptoms now that you've been introduced to the pathogen and the vector. I have to make a brief point here that scorch is actually a symptom. So the symptom we see where the margins of the plant become yellow or chlorotic and start to turn brown or necrotic and start drying out. So that is a symptom. The cause of the scorch can range. This is the issue we have with symptoms, is many different causes can result in the same symptom. I often equate this to how many different things can cause a headache. Seeing a symptom like scorch would be much the same. So causes of the scorch can include drought, which also means drought as a result of frozen soils. If you're on the east side of Washington, we see this quite frequently. Our conifers experience this type of scorch. Salt damage can also result in scorch. So salt from de-icing salts or even pet damage can cause a burning of the margin as the plant is trying to get rid of the salt through the edge of its leaves. Nutrient issues, both deficiencies and toxicities, can look similar. Air pollution. Now this can also be air pollution as a result of wildfires, and we start seeing this quite frequently in eastern Washington, the Columbia Basin, well actually probably all of Washington, where during those heavy smoke covers we actually see the plants respond in a leaf scorch symptom. So those were our abiotic causes. We also have the biotic causes such as fungi, bacteria, even nematodes, things we normally think of being in the ground, can cause leaf scorch, as well as viruses. So again, leaf scorch is just going to be a symptom as a result of water. So to understand that a little bit more, how a plant loses water is a result of how water moves within the plant. So the plant will take up water from its roots, and it is actually passively lost up through the stem and through the various branches into the canopy, where it's then lost to the atmosphere. If you think about it, if all of us remember back to our early physics, early chemistry, we're going to be moving from areas of high concentration, aka our plant, to areas of low concentration, the atmosphere around it. This loss of water can be a result of, heat. A lot of light can cause increased loss of water, whereas the addition of water into the soil helps reduce these drought symptoms, these scorch symptoms, and again, sometimes cloudy weather will also lessen the impact of drought. Now, the way I talk about it, if you've ever heard me give talks before when I'm explaining how the vascular system works in a plant, is it's much like if you had a bunch of straws that you're trying to drink soda or juice with. With these bunch of straws, you could suck up all of your soda or all of your juice, and then if there's no more, right, you go into a drought-like symptom. You've lost all of the water, there's no replacement. Now, what we start to see with particularly pathogens, even insects, if I were to come along and start clogging a couple of those straws, let's say you have 10 and I clog 2 with wads of napkin, you've now reduced how much liquid you're getting into your mouth, right? But you're still getting some amount of liquid. So this is what happens when we start clogging the xylem vessels with pathogens, especially those that are growing into those vessels. They start clogging that area. Now, that doesn't mean all vessels are affected. You know, if I've clogged these 2, the rest of these still work fine. So there is some water getting up into the plant. But those areas of the plant, say a branch, that might be relying on this clogged straw are going to start showing the symptoms of scorch. Anytime I give a presentation, you're almost guaranteed to get really terrible clip art. So here you are, you're welcome for that clip art. So keep this in mind as we start talking about the symptoms that we see. Okay, so scorch symptoms, when it comes to Xylella, we often see a sharp line between the healthy and affected tissue. This is what we're talking about here. There's a really fairly dark border between this dead tissue and the parts of the tissue that have not yet been affected. Some of this yellowing and redding have to do with the seasonal change. But in these other photos, you can see this dark line between that dead necrotic tissue and the tissue that is still. We will often see an intervenal chlorosis or necrosis, again, that yellowing or that dead or brown tissue. This has to do with the fact that the veins are going to be carrying the water, so they tend to retain that moisture longer. So the tissue at the margins, working in, those will be affected first. So we tend to stay green near the veins. Now think back to our really terrible clip art example of soda straws. When we look into the canopy of a tree, we should see an irregular distribution, as only certain branches are the ones not getting the water. So we will only see certain limbs, maybe even certain parts of a single limb showing these symptoms of scorch, these symptoms of intervenal chlorosis. Now, as the xylem continues to be clogged, we will eventually start to see some leaf wilting, probably leaf drop, eventually dieback, particularly on those affected branches, and then more branches will start to be affected, and we can eventually see death. Now, it is important to note that different species and different strains, so different host species and the different strains of the pathogens can result in different types of symptoms. When I was introducing Pierce's disease and phony peach disease, so this is again the same pathogen, but one was causing scorch and one was causing stunting. So we have to keep in mind that these symptoms can change, again, depending on which species. I do just recommend getting to know your plants, expecting what is normal, what might be seasonal drought, and just keeping an eye on how the plant is doing, and especially noting those canopy symptoms, because again, drought should be affecting the whole plant. That's the same thing that happens when you drink all of your soda out, right? No, none of those straws have any liquid left versus that clogging. So again, hopefully my bad clip art has served its purpose in lodging this in your memory. Definitely, we have a lot of lookalikes. Our abiotic lookalikes can be things such as drought. So here we have maple or sycamores, both affected with drought. Now, again, drought's going to cause the same symptom as scorch, bacterial leaf scorch, because the same process has been interrupted. So here we have this plant sample, or again, we see intervenal chlorosis. You can actually really nicely see the veins and then the responding areas of brown as well as yellow. And this was drought. The entire tree was impacted. They ended up having to fix the irrigation system, and then the tree recovered. So there's a lot of mimicking in the symptoms because, again, the same process has been impacted. Now, if you look at the whole tree as we see here, again, we should see a more uniform pattern of damage on a tree that is drought stressed because, again, there's no water to be taken up, so the entire tree is impacted. Now, with salt damage, we can see a similar kind of interveinal chlorosis and burning along the margin edge. But again, most of the plant should have some degree of symptom if it is an abiotic issue. But just know that people like myself and Jenny Glass over in the Puyallup Research and Extension Center exist to help you figure these things out. Now, another lookalike is something that I think a lot of us come in contact with. This is verticillium wilt. It is a big issue on a number of our urban trees. Verticillium is another pathogen. It is a fungal pathogen this time, however, and it's going to create really similar symptoms because to Xylella, and that is again because it is clogging the water and nutrient conducting tissues within the plant. So we do again see interveinal chlorosis and necrosis, that browning. We can see flagging, so individual limbs and leaves, not a consistent pattern across the entire canopy showing up. And again, verticillium wilt can be quite problematic because over 300 species of dicotyledonous plants can be impacted by this particular fungus. So this is another pathogen with a really big host range. Now, in terms of trying to figure out if you have verticillium wilt versus Xylella, a really nice thing about verticillium is that on some of the affected branches, especially if they're a bit larger, say about the size of your finger or your thumb, if you cut into the tissue, you will actually see kind of a green to olive to brown discoloration inside of the vascular tissue. What we're looking at is actually microsclerotia. This is a fungal structure that the verticillium uses to survive long term. So if you're working and you prune a branch that you think might be infested, you could see this. You can also take a knife and cut through, and you should see some discoloration in the tissue. Now, this is not always present, especially if it's smaller diameter tissue, if it's an early stage of infection, because these microsclerotia are forming in dying branches in the hopes that they'll make it down to the soil, where once that plant tissue decomposes, they'll be released and able to cause new infections. So clinics, again, like myself and Jenny Glass's in Puyallup, we test for this routinely. So it's a really pretty quick test to turn around. So we can detect it even if these microsclerotia are not present in to be seen with the naked eye. Some clinics, including mine, can also test soil for this particular pathogen. So if you're looking around trying to determine whether or not you have Xylella or verticillium wilt, it is worth sending into the clinic, and we can quickly identify if verticillium is involved. So next lookalike is anthracnose. Anthracnose is an umbrella term for a number of fungi that attack many of our trees. Again, they have a very wide host range. There's a number of different fungi involved. They do cause chlorosis and necrosis on leaves that is frequently associated with veins that can easily be confused with bacterial leaf spot. A nice thing about anthracnose is it tends to be more of a problem earlier in the spring when we have more wet and mild conditions, which would be the opposite of leaf scorch, which will be more prominent as far as symptoms when we have some more stressful conditions on the plant. With anthracnose, again, this can be checked for in the clinic very quickly, as well as if you see fungal fruiting bodies like we're starting to see on this particular leaf, that would be a good clue that you're dealing with anthracnose versus bacterial leaf spot. So how the disease cycle functions for Xylella fastidiosa bacterial leaf scorch is what happens is, so we have an organism, a plant that is infected with the Xylella fastidiosa bacteria. A piercing sucking insect, likely sharpshooter, feeds on the xylem tissue and it takes up the bacteria. So then this infected leafhopper then moves to a tree host, feeds on the tree, was able to make contact with the xylem, allowing the bacteria to then be introduced to that area. That bacteria then reproduces to the point where it starts clogging the xylem vessels, and that's when we start to see symptoms. So as more and more of those xylem vessels begin to clog, we start to see more symptoms start to develop. So as you can see here, here's what we expect our healthy tree. This dark gray is supposed to represent dark green. So we would see canopy symptoms that are irregular, different branches more impacted than another, different tissues. And now this is able to actually start that cycle again because this tree is now a reservoir for this pathogen. So with Xylella fastidiosa, what is the big deal? The big deal is this pathogen can move very easily. So the insect vector can move itself. They are able to fly, but they only fly very short distances. So this is a key thing that we've figured out, that the movement of the insect vector by itself, unaided, is actually very small. It does not move very far from the host that it's feeding on. However, weather can sometimes physically move a small insect. These are little tiny things. As well as weather can start changing as we see differences in temperatures becoming more regular. These vectors are able to increase where they're able to live. So we can see the spread of the disease as the insect vector itself is able to start existing in what potentially used to be a harsh environment for it. Humans, we're really good at moving pathogens and vectors, unfortunately. So humans can accidentally introduce organisms. This is what happened with the glassy wing sharpshooter in the case of Pierce's disease. We accidentally introduced the sharpshooter into the area. Infected plants. This has actually been our major pathway for the movement of Xylella fastidiosa. What happens is we move an infested plant to an area through shipping, commercial trade, and we talked about the fact that these potential vectors are present across the globe. So in the EU, if infected plant material got its hold in the EU because the plant was introduced, an infected plant was introduced, and then the spittlebugs were able to move it to other uninfected plants, thereby continuing that life cycle of movement throughout the area. So how do we detect this pathogen? I did mention we can't culture. Some labs that work on this pathogen might find it worthwhile to use this media, but it's not very friendly. Again, it's a picky eater. It doesn't like to exist unless it's in perfect conditions. And it's very hard to replicate the inside of a tree inside of a lab. Now, this pathogen is not always detectable. When surveys are being conducted for this particular organism, surveys have been pretty extensive in Washington, Oregon, as well as other areas in the Northwest, trying to find this pathogen, see where it is. But it has to be done when the tree is active. So this tends to mean spring through early fall is the best time for us to actually detect this pathogen, because we have to have enough of the pathogen present. We often call this titer, so enough pathogen present that we can actually detect it. So there is a level of seasonalness to this pathogen. One of the methods used was actually an antibody detection method. We call this ELISA for immunosorbent linked or enzyme linked immunosorbent assay. I knew I got a letter wrong there. Unfortunately, this hasn't been as robust as we hoped. Part of that is due to the fact that you need quite a bit of bacteria in the affected plant tissue for the antibody test to actually detect it. So the gold standard now for detection is using molecular. So we use DNA, or use PCR to detect the DNA. This also allows us to identify which strain of the pathogen is present. Now, this is very important as different strains are more or less concerning, especially in the Americas, where we do have some level of endemic strain to us. Now, management. So the past two trees I've shown have hopefully striken you as these would be trees you'd want to sample, being that you have some areas that are browning, necrotic, probably a little chlorotic, some thin areas, as well as parts of the tree that look very healthy. So again, we need to confirm the cause. This is because management can be drastically different. If this happens to be a drought-related stress, well, you're not going to want to spend a ton of time pruning when you should have been putting time towards watering. And if it's verticillium, your practices to prune out affected tissue would be drastically different. The other big thing with, if you have a tree you suspect has Xylella, is to get it tested, because we want to ensure that the strain of Xylella, I mean, let's hope that it's not Xylella, but the big thing is we want to confirm which strain is present. Again, Xylella fastidiosa has likely been present in the Americas, so North, Central, and South, for quite some time. But these strains, that's where we see different levels of invasiveness. So we want to make sure it's not a new strain to our area, or a strain that can threaten other plants, particularly, our various industries, our agricultural industries, that could definitely be very negatively impacted. So with Xylella and other things that damage vascular parts of trees, we want to practice good growing behaviors. So avoiding stress, avoid drought stress, heat stress. This also includes reflected light, because that will encourage more water to be lost. Reducing any root or trunk damage, because if we're already compromised in our vascular tissue, we definitely don't want to add on any more root damage. And then soil issues, so compaction, so the packing in of dirt, as well as drainage issues. So this seems counterintuitive. We want our soil to drain adequately, because if our soil stays too wet for too long, we actually can see an anaerobic rot. So the water starts replacing the oxygen in the soil, and that in turn starts damaging the roots, so they're no longer functioning as they should. Careful fertilization, we want to make sure it's balanced and encouraging good root growth and not just top growth. This is why I always seem to pick on nitrogen, so my apologies, nitrogen, but nitrogen encourages nice flush top growth. That puts a lot of stress on the vascular system, so we have to be careful with how we fertilize. The use of tolerant plants, so those plants that the bacteria doesn't tend to reproduce in, and then avoiding susceptible plants. So if we know certain plants are more susceptible to present strains, maybe avoiding those. Big thing is also to remove affected plants and potential pathogen harbors. So again, we have those asymptomatic hosts that can keep the pathogen present. I mentioned that this is a vector pathogen, but unfortunately, vector control isn't really practical. They haven't shown really great results in trying to manage for the vector. Again, it doesn't move very far on its own, so control at that level can be difficult. All right, so that was Xylella fastidiosa. Now we're going to move on to our second tree threat of the morning. We're going to talk about borers. So this is my new favorite photo. In general, it shows a number of the things that attack trees, one of which is borers. So borers are insects that literally bore or kind of burrow their way into a tree or plant. They do attack non-woody vegetation. And then something I was reading said, or rock, and I'm no entomologist, but that seemed very odd. So if I can have an entomologist confirm that they do truly bore into a rock for part of their life cycle. Now, usually an adult will bore into the tree, some woody tissue portion of the plant. It'll lay eggs, and the larva will spend part of its time eating on the plant. Now, this is very traumatic for the plant. As you can imagine, there's an insect boring its way through. So to go back to our soda and straw example, this would be like me coming around and poking holes in your straws. I'm quite literally damaging those tissues you need to get your soda. So borers do have a role in the ecosystem. I think this is important to make note of. Borers help speed up the death and decline of trees, but they're doing so on weak trees. That is their goal. They want the easy food. Trees, these are particularly drought stressed, older trees. They might be impaired. They might have other damage done to them at the time. This is because those stressed out trees actually release a chemical that these borers can usually detect, and they will intentionally target those trees. So the benefit in the ecosystem is that a new tree gets to grow in that unoccupied space. So the borers we are typically concerned about are those that are attacking relatively healthy trees. So we have primary versus secondary invaders. So a primary invader is a pest that is attacking a healthy tree. It is important to note that a healthy tree can actually withstand minor damage from a primary invader. Trees do have the ability to produce sap and resin and other materials that actually kind of clog up those damaged tissues. So they are repairing those straws, if you will, so that it can function normally again. So these are much less of problematic issue for trees. Now, a secondary invader, these are the ones we talked about with the role in the ecosystem. They're those that attack trees that are already stressed, and they're there to help contribute to the decline and death of those stressed trees. Again, playing a role in their own ecosystem. So we have different groups of wood-boring insects. The first are beetles, such as the group Pestid. These are usually flathead borers, so their larva is actually a different shape of head. We have the Cerambycidae. I am so sorry if there's entomologists who are cringing at how I'm pronouncing these Latin family names, but they are often our round-headed borers, such as Asian longhorn beetle. Now, please note that these top two organisms are actually incredibly concerning invasive pests. So this is emerald ash borer and Asian longhorn beetle, and we'll talk a little bit more about them, but we definitely don't want them in our area. They're quite devastating. So it's not just beetles that are capable of boring. We also have moths, such as clearing moths and carpenter worms, as well as wasps, such as horntail wasps. So lots of different organisms can actually be wood-boring insects. Now, there's many times where we don't actually catch the insect in the act, if you will. So we're going to have to be using different parts of the plant to figure out what pest might be present. Now, it's important to pay attention to what part of the plant is targeted. So is it the xylem, phloem, cambium, or bark? So here we have two different examples depending on how you spatially orient yourself when thinking about trees. So, because we can have phloem feeders versus xylem feeders. So a phloem feeder is going to stay much shallower because it's only feeding in that bark to the cambium layer. So it stays shallow, so we see the galleries are kind of, I think they look like racetracks, left behind from phloem feeders in a very shallow level. Whereas with things like Asian longhorn beetle, they are able to burrow all the way through to the xylem part of the plants. I mean, they can quite literally cut their way. Clearwing moth is the same. They can leave really large holes. This would definitely kill a tree very, very quickly. My personal favorite organism is walnut twig beetle. I probably like this because it plays a role in thousand cankers disease. So this is a very shallow feeder. It stays only in that phloem area, so just barely under the cambium. So this is an example of, it's important to know what organism you're dealing with, because when you're checking, I definitely make knife cuts into trees much further down into the vascular tissue than this. So you should start shallow when you're doing your examinations to see what organism might be present. So these borers do have some levels of host specificity. So emerald ash borer, as the name implies, is host specific. It targets ash species. Now, because scientists do odd things, sometimes we confusingly name organisms. Red-headed ash borer is actually a generalist, even though the word ash is in the name. Bronze birch borer, something we definitely have in eastern Washington, is host specific. It goes after the Betulaceae family of plants, whereas Asian longhorn beetle is another generalist. So again, keep in mind the emerald ash borer and our Asian longhorn beetle are our big bads in this group. We definitely don't want them, particularly since Asian longhorn beetle is such a generalist. So signs and symptoms of a borer infestation, these include things like exit holes, oviposition sites. Sometimes we can actually see the various life stages of these organisms. But we'll also see some tree-based damage, such as wilting, thinning, or canopy dieback. There's a lot of symptoms, so it's important to start learning the symptoms and signs associated with borer infestation, because again, we don't often get to see the life stages. We just see the damage that is left behind. So first are exit holes. So exit holes are quite literally the door to the plant, how the insect is moving in and out. Now, depending on the species, these exit holes will look different. So with our flat-headed borers, we tend to see a D-shaped or a capital letter D-shaped exit hole because of, again, the way its head is shaped produces this particular exit hole. We can have some round exit holes. This is the case with shot hole borers. And then the size can also be drastically different. Asian longhorn beetle is about the size and diameter of a number 2 pencil, whereas walnut twig beetle, the exit holes are about the tip, just the tip of a mechanical pencil. Again, here's an example. So a round-headed borer leaves a round hole. A flat-headed borer leaves a D-shaped exit hole. Frass and sawdust are the other 2 signs. So we can see insects kick out the frass and the mix of sawdust. So frass is actually the excrement of this boring insect. And so what we can sometimes see is what we call a frass tube. So this is a mix of both frass and sawdust, or the sawdust of that woody material. So that's what we're looking at here, where it looks like there's little tiny twigs coming out of this tree, but it's actually getting removed by the organism and mixing with its frass, so it creates these tubes. So with the sawdust, again, this is the plant material that has been removed so that our borer can have room inside of the plant. We often see it either on surrounding the base of the plant, or especially in these kind of crooks of the plants where we have branches meeting that main trunk axis. So those are frass and sawdust. Next are oviposition sites. So these are where eggs are being laid. So this can come in kind of 2 forms. So with Asian longhorn beetle, we actually see chewing left behind on the bark. So they're kind of clearing out a little niche for their eggs, where they will then lay their egg masses. Now these egg masses, in this particular situation, the bark of this tree is more white and gray, and the egg mass is more amber. Now on this tree, that would be much harder to see. So sometimes these oviposition sites and egg masses will change in colors as they age. So depending on when you can catch them will dictate how easy it is to actually see these oviposition sites. Because you can see in this photo, they can be quite small. With a good hand lens, though, you should be able to see these clear notches from Asian longhorn beetle. Next is canopy symptoms. So again, this would be what most of us are, this would be what would alert us to something going on with the tree. So this yellowing, thinning, often a top-down dieback that tends to be quite typical for borer pests. Because again, we're interrupting the flow of nutrients. This goes back to the same thing with the straws and our soda, but it can be quite damaging. So by the time, hopefully someone would have noticed these symptoms before it got this bad, but by the time we start seeing canopy symptoms that really alert us to these trees, these are walnuts affected by thousand cankers disease, which is vectored by the walnut twig beetle. By the time we see these types of symptoms, that tree will be gone in 2 to 3 years. So that is a sobering fact to keep in mind with these borers, especially those that are invasive and attacking healthy trees, otherwise healthy trees. So woodpecker activity is next. Woodpeckers and sapsuckers that feed on the trees are actually looking for the larvae. So they're taking their beaks, digging into the tree, trying to find the larvae for them to snack on. What's important to note is, I will own up, I made my first mistake with mistaking woodpecker activity for borer activity 5 years ago. But one key to notice with woodpecker and sapsucker damage versus an exit hole is the birds tend to be quite linear in how they are pecking these holes, whereas the insects are going to be more random throughout a tree. So if you think about it, the woodpecker is going to be tapping here, here, then moving down, tapping again, trying to find that larva for it to snack on. Now, another big issue with the pest activity is we start to have integrity issues. So as that pest is feeding through the vascular tissue of the plant, that tree is becoming weaker. So if we have windstorms or freeze damage, the tree is actually more likely to break, as this poor car found out, unfortunately. Next are epicormic shoots and bark splits. So epicormic shoots are a stress response from trees where during a stressful event, they'll actually send out more shoots, hopefully trying to get more green tissue for photosynthesis. So they're responding to the borer damage at that point. And then bark splits. For something like Asian longhorn beetle, we tend to see bark splitting as the tree is trying to continue to grow around that damaged material. So these are other symptoms of these insect invasions. So speaking of invasions, we do have a handful of, oh, one of my pictures didn't show up, sorry. We do have a handful of invasive diseases that we are quite concerned about. Hopefully most folks are familiar with emerald ash borer and Asian longhorn beetle. These are organisms that are present on the east side of the country. But one of the things that is present on the west side of the country, in our home state, depending on if you all are from Washington or the West Coast, is thousand cankers disease. It is a combination of a boring insect and then the fungus it leaves behind. So this again is my favorite little guy, walnut twig beetle. So these are teeny tiny organisms. These were the ones that their exit hole was about the size of a mechanical pencil tip, and we can fit 10 of them on Abe Lincoln's head. They each carry with them fungal spores. So for every canker, so every entry and exit hole, they introduce fungi. That's what we're actually seeing growing in the wood right here. So the beetle doesn't actually cause all that much damage itself, other than it signals to everyone else in the vicinity, hey, I found a tree for us to chew on, come hang out. But it's the fungus that it leaves behind that really impacts the tree. And I might be biased because I am a pathologist, not an entomologist. So for detection and identification, luckily we have two groups that we can use to try and figure out what we're looking at. So first is the larva. Sorry to do this right before lunch. What we can look at when we're looking at the larva is the shape of its head and even the segments. So for emerald ash borer, it has very bell-shaped segments, and that can be very distinct for this species. Also appendages, that's what we're looking at down here on the, I think I'm allowed to say, tail end of the organism. Those little appendages can also be very diagnostic, as well as the galleries they leave behind. I referred to them earlier as racetracks, but they're the area of the tree that have been fed upon by the insect, and this can be distinctive for the particular pest. Different pests have different patterns of the galleries. For example, walnut twig beetle, the larvae feed parallel to the wood green, and the adults feed perpendicularly. So that type of information helps us figure out what organism might have been present. Now, with the adults, if we happen to catch the adults, the size of the adult, the shape of the adult, and its feeding behavior, again, if it's a xylem versus phloem feeder, can be very helpful, as well as how its oviposition site behavior can also be very helpful. Again, emerald ash borer is in some ways nice because we can easily identify it based on its very distinctive metallic green color. But just like plant diseases, we have many similar organisms that we have, lookalikes that we have to deal with. So with management for different borers, again, you need to confirm the cause because your management options can vary. Different pests are going to be responded to differently. Here we have bronze birch borer. Now, if you have just one birch that is affected, it's probably a birch that was already stressed out. So we wouldn't really recommend you responding using chemicals or pesticides, or excuse me, pesticides. And we also need to respond to an invasive pest. So if you're seeing damage and you have a reason to believe that it could be one of our invasives, it's important to make sure a sample gets sent in to a clinic, to Extension, to WS or Washington State Department of Ag. That way we can respond if we need to. With our borers, again, most of them are going after stressed plants. So healthy plants will resist borer infestations or keep them from moving into an area. So this means proper pruning at the proper time, watering, and appropriate fertilizing. Minimize stress. This also does include sunburn. I know a lot of our fruit trees start to get borer issues, shot hole borers, especially if they have sunburnt areas. Sometimes you might need to remove the tree or the affected tissue. This does include actually any dead woody tissue nearby, because again, these are organisms that like to feed on dead wood, especially if they are secondary invaders. So removing that from an area can help protect your healthy trees from that minor feeding damage. There are different pesticides. These must, I'm gonna make my language a little stronger there, these must be used per label instructions. Your label is the law. You cannot use a pesticide in any way that is not consistent with its label. If you want more information about the different pesticides that are available, the Pacific Northwest Handbooks has a nice little flyer on different pesticide options for these. So the biggest thing with tree threats is if you see something, say something. I stole that from TSA, but it works for us too in the plant world. A big thing is if you're unsure of what to do or what you need, call the clinic. Call myself. You can call your local clinic, local extension, and we can guide you on what type of sample you can take. With my wheat growers, I tell them to send me a whole plant. None of you are going to send me a whole plant, or at least please don't send a 6-year-old tree. That is very hard to, don't quite have the space. So I can help you figure out what type of sample you need. Sometimes we can start with photos to help determine what's going on. And if necessary, I can connect you directly with the resource you need. If this is a case where I think we really need a WSDA inspector, I can do that part of the communication and notifying them and getting you the help and resource you need. Big thing is just make sure you contact someone at the extension, the university, a state program. You can also use our Invasives reporting app, because the biggest thing here, especially when we're talking about invasive species, is early detection is critical. Submitting samples, I say submit a sample that you think is infected with an invasive species, treat it like a dirty diaper. If you had to take a dirty diaper with you or put it in the mail, you'd probably want to make sure it didn't come in contact with anything else, right? We don't want to have to deal with that. So double bagging, ensuring that it's in a nice sturdy container, well managed, is really important. So again, whole plants are probably not likely for most of us. Affected branches, a ½ inch to 1 inch in diameter, not dead. Dead things don't give us good answers. When it comes to insects, as many life stages as can be found is important. Sturdy packing material is also important. No one likes goo, so we have to try and prevent goo from happening. If it's on plant material, you can go ahead and collect the plant material, put that in a sealable plastic bag, and then it can be sent as is. Flying insects may need to be killed overnight by putting them in the freezer. You can also put them in alcohol, but be sure to remove the alcohol prior to sending, because 1, it's quite concerning to get an unknown liquid in the mail, and 2, it's illegal to put alcohol through the mail. If you find larvae should be sent alive, because an entomologist is usually going to have to rear or grow that larva to adulthood to actually identify what it is. So what to do if you suspect an invasive species? Download the Washington Invasive Species mobile app. It'll allow you to take photos, and then those photos are directly routed to an identifier. Contact local extension, contact a diagnostician, submit a physical sample. This is important because think how many symptoms looked the same, you know, from our diseases to abiotic to our borers. And then just a word to the wise, be cautious with the information. If you suspect that you have one of these invasives, let, you know, Washington State University, Washington State Department of Ag, and the United States Department of Ag, let us work to make sure that we do in what the identity of that organism is. Because if you get excited and start going, I have emerald ash borer on Facebook, I don't know who would post that, but that was the only example I could think of. That could trigger a lot of panicked responses. So be cautious with the information. Let us have time to confirm what the problem is, and then, you know, we're here to help. Hopefully none of the invasives happen, but just keep that in mind. There are different diagnostic clinics available to us in the Pacific Northwest. We are very lucky that we have 5. Please ignore this email. I will update that. With that, I'll take any questions.
Awesome. Thank you so much, Rachel. That was excellent. And like Rachel said, we're going to open it up for a little Q&A. Rachel, we do technically have 2 minutes left, but
Okay.
Are you okay going a little bit over?
I am.
Okay. So for folks on the webinar, again, you know, if you want to leave at 11:30, that's fine. Any questions that Rachel answers now will be recorded in the video we're going to upload. And any questions that we don't get to in, say, the next 5 minutes, we are going to answer via email and send out to the group. So you will not miss out. All right. So just lost my question board. Sorry, 1 second. Well, one of the questions here is dripping pitch an indication of pine borer?
That is a great question. The answer is yes. So dripping pitch or sap from a tree is an indication that you could have a borer present. Now, we do have different diseases. It will depend on the plant because there are different diseases that would cause pitch or sap to drip out, even to bubble up. But that is a concerning symptom and worth looking into. So yes, if you are seeing pitch, I do recommend looking into making sure you understand what is causing that on your tree. Great question. Sorry, I didn't mention that.
No worries. So another good question. When you see a tree expressing scorch symptoms, does the time of year the damage develops tell us anything about what may be causing the scorch?
That is a good question. Yes. So the time of year in regards to when we see scorch symptoms can help us figure it out. If it's in the summer and we've had a long time in between rains, we can expect to see more scorch than we normally would. And same thing coming out of winter, if you're in an area where the ground freezes, you might see more scorch symptoms because the tree hasn't been able to take up water. Now, in terms of the pathogen, that scorch symptoms, that would be less seasonally, it's hard to say. That one can be a little less seasonal in the fact that you could see it in spring, you could see it in summer, and you could see it in fall because it's physically blocking. There might be access to water, but it is blocking the water. It will probably be leaf scorch caused by bacteria will be more pronounced during drought stress because of the increased drought stress. So hopefully that made sense. But yes, it can help us diagnose.
All right. Let's see. Can you talk a little bit about the different Xylella fastidiosa strains, the ones common to our area and those being imported from other places?
Sure, that is, I can tell you what I know. That is kind of a loaded question. We are figuring more of that out. There's a great website hosted by UC Berkeley, the Madeira Lab. So if this person wants to contact me directly, I can actually put you in, get you better courses. So we do have 6 strains. So subspecies fastidiosa is the Pierce's disease strain. That has not been found in the Northwest so far from all of the surveys I'm aware of. In the southern part of the US, we do have a subspecies called sandy eye. This is what's attacking a lot of pecans and oleanders. And then the multiplex subspecies is often on almonds and can also be found on some of our shade trees. So.
All right, so thanks for that answer, and we'll include that link when we send out the answer to the other questions, if that's possible.
Absolutely.
Yeah. All right. Let's see. We'll take, I think for sake of time, we'll take one more question, and again, all of these will be answered and sent out to the group. What tree species are resistant to scorch?
That is a good question. I don't know if we have trees that are truly resistant. Resistant means that the bacteria is not reproducing or unable to really cause any damage to it. I'd have to look into that. So if this asker wants to email me, I know we do have some weedier hosts that are means the bacteria is in there, probably reproducing at a low level, but not really impacted. But again, please send me an email and I'll get you more concrete information.
All right, excellent. Thank you. And I just wanted to put a quick plug for people on this webinar. So this webinar is part of our overall program known as the Washington Pest Watch program. And it's a citizen science initiative led by agencies and universities that are on the front line of protecting our state's natural resources and economy from damaging effects of invasive species. And most of the people in this network are everyday people who just keep their eyes open for signs and symptoms like the ones you learned about today, and making reports to us. So Rachel did a great job. Sorry, I'm plugging the Washington Invasives app. It's a great way to just take a picture, send in the location, and we'll send it off to whoever is the expert in that field. So if you see something, say something. And we have a list of resources on our website highlighted here on the screen. We have past webinars that have been recorded. We have pre-recorded trainings that you could play for a group, maybe if you're a master gardener or something like that. First Detector Handbook, we just have a lot of great resources on there if you're interested in helping out preventing invasive species and protecting your plants. Thank you again to Rachel for that incredible presentation, and thanks everyone for joining us.